A very good day, my dear undergraduate students, both BDS and MBBS. The topics on the screen are a little abstract and seemingly vague, but definitely important, particularly the first two topics. The references are given over here. R.A.T.S is my favorite. Your book should be yours. Robbins, of course, is a backbone. First one shall be amyloidosis. Amyloidosis is a process and amyloid is a protein. Actually, the word amyloid means starch-like. That is what Rudolf Virchow had thought. But then it turned out to be a protein. Under that, you will be having what is the nature of the amyloid. For example, in the book, there is a physical nature of amyloid. It is a never-ending fibril, about 7.5 to 10 nanomicron. See whether you are able to find that out. And it can be identified by means of spectroscopy. And the chemical nature can be, it is composed of fibril proteins and a P component. These are all important. See that they are given in your book. And next will be the classification of amyloid. There are different ways of classifying. Earlier, it used to be primary amyloidosis, secondary amyloidosis. But now, there are so many conditions which are associated. Therefore, the classification will be needed. For example, what is AL amyloid? Light chain. So, what is the precursor for it? AA amyloid, transthyretin, beta 2 microglobulin. You can get questions like, which of this is associated with renal dialysis? Which of this is associated with myeloma? So, there can be a catch in it. Having said that, next can be the pathogenesis. So, in that one, you will be having the various steps. For example, what is the monoclonal B lymphocyte, the plasma cell, immunoglobulin, and then you get a amyloid light chain. Or there can be a mutation, transthyretin and ATR protein. Similarly, there can be inflammation which can be leading to AA or amyloid associated. This particular flow diagram and the tabular column that I mentioned regarding the types of amyloid are mandate. 50% of the mark goes for this. Do not write primary and secondary, that is obsolete. The stains for amyloid. So the stain will be starting with iodine. What are all the various stains that can be used? For example, I have got Congo red, fluorescent stain. Then PAs can be used. Alcian blue gives a blue color. There are some stains called as metachromatic stains. So the substance will take a different color. The background will take the color of the stain. That is why it is called metachromatic. Under that, you have got a toluidine blue. Similarly, you will be having certain things like the amyloid T. So I would like you people to kindly remember all this. Thioflavin T, thioflavin S. They are all the fluorescent stains which you will have to remember. The stains for amyloid is a definite one. It can be asked as a short note. It can be part of an MCQ, part of an essay itself. What are the various organ changes in amyloid? Particularly in this context, amyloid liver, amyloid spleen can be asked as separate questions. The sago spleen, lardaceous spleen, amyloid in the place of disease, in the case of liver. So what is the appearance that you people get? The clinical features, it can be like a tumor presenting. That is called as tumor amyloid when the tongue is getting enlarged. Or it can be associated with malnutrition. Or sometimes it can be associated with malignancy. And the diagnosis, apart from the special stains, you'll have to mention the histological picture, at least in the biopsy. And the light chain, or rather the light microscopy, amyloid means it is a homogeneous, eosinophilic, acellular material. Therefore, only you are doing all the special stains to identify it. Sometimes in organs such as the heart, it can lead to defect in the conduction system. The patient can go in for a cardiac arrest. So amyloidosis is a vast topic. You people will have to break that up. And even if you read one component per day and by heart it, it is good enough. We can always repeat it and consolidate. The next one is the HIV, AIDS. It was once a frightening disease, much more than COVID-19. Now probably people have forgotten that. Anyway, we'll do it. It is an acquired immunodeficiency syndrome. That is why it is called AIDS. The routes of spread. Commonly, it is a hematogenous spread. It can occur through pregnancy and a sexual intercourse. These are all the things. Accidental, particularly iatrogenic, in doctors and nurses, it has been common. The morphology is, it is a retrovirus. And if you look at the book, there will be a spherical structure along with a glycoprotein in the surface. This particular diagram you people will have to draw. So there is a glycoprotein GP120, GP41, etc. And what is the structure of the virus? It is a question definitely in microbiology. In pathology also we will mention it to that extent. Now, how does this virus survive? No virus can survive independently in our blood. So what happens is there will be a diagram. Just like phagocytosis, it gets engulfed and it gets attached, it is engulfed, and then the viral protein gets incorporated into the host human genome. There, it uses the host DNA for its multiplication. And after that, you find again it is converted to the RNA. It makes use of the host tissue to form a cell membrane and it gets released to be engulfed by another cell. That is why you find progressively all the lymphocytes and the macrophages will be diminished. So you get a recurrent infection. In fact, multiple infections can be occurring in these patients. What are all the infections? It can be tuberculosis, it can be opportunistic fungi, and sometimes there can be some tumors such as the B lymphomas which can be associated with it. So when a question comes regarding the HIV, you people will have to write what is the acquired immunodeficiency syndrome? What are all the routes of infection? What is the structure of the virus? And what can be the methods of diagnosis? That again you people will have to mention. It can be an ELISA, it can be a Western blot. Sometimes we use a simple technique called the tri-dot method, PCR, polymerase chain reaction. These things you people will have to mention. The neoplasm, Kaposi sarcoma is one of the most common ones. The mode of engulfment, etc., you people will have to describe with a sketch. So also the structure of the virus. I hope this particular topic is clear for you. The next one can be a rejection, a graft rejection. Generally, a transplant is required. You people can give a couple of examples: the bone marrow transplantation, renal transplant, etc. And in all this, there can be a rejection. That is because the body is not accepting it. And it can be an acute rejection or it can be a chronic rejection. So see whether you people are able to locate it. Sometimes graft rejection rarely can be asked as a question. And what is a graft versus host disease? So there are the T lymphocytes which are attacking it and ultimately they make it incapable. Sometimes there can be a hyperimmune reaction also. Acute graft versus host disease. The graft will be attacking the host. So these two things you people will have to write. Graft rejection, HIV, and amyloidosis. I hope that is bringing us to the end of this class.